Good afternoon. Today the topic of discussion is about the structure and the function of bacteria. Today is the first class, so I will be discussing about the introductory parts of microbiology and the following topics. As per I have shared you the routine, your classes will be held accordingly and with time. So the first is the structure and the function of the bacteria. You need to know details about these things because it will help you in understanding the future aspects of how bacterial structure, functions, etc. do takes place uh, in cases of any systemic diseases as we proceed on to the other topics. So first of all, I will be making you familiar with some bacteria names we often use. So these are Cocci, Bacilli, Vibrio, Spirilla, Spirochete, Actinomycetes, and Mycoplasma. The individual characteristics of these are whenever we will be referring to cocci, we will be meaning some spherical and oval shaped bacterial cells. The bacilli will be indicating rod shaped cells. Vibrio are comma shaped curved rods, and Vibrio will be dealt separately. Spirilla means rigid spiral forms of bacteria. Spirochetes are flexuous spiral forms. It's just like a coil. You have seen a spring in pen and etc. So it appears just like that. Actinomycetes, it's a branching filamentous bacteria. Actinomycetes will be dealt later on. And Mycoplasma, these are the bacterial forms which do not possess cell wall. As it lacks cell wall, it does not possess any special cell. Okay, so Mycoplasma will not be dealt with separately later, but just remember the thing that whenever we will be referring to the term as Mycoplasma or giving any example of Mycoplasma, that indicates that the bacterial that it is the particular kind of bacteria which do not possess a cell wall and hence does not has a fixed shape. Now about the structure of a bacterial cell, what does it consist of? It consists of cell wall, cytoplasmic membrane, cytoplasm, nucleus, slime layer and capsule, flagella, fimbriae, and spores. We'll be discussing about the individual structures of the bacterial cell component. The first is the cell wall, which is the most important part of bacterial cell. It gives the bacterial cell its shape. It cannot be seen by direct light microscopy. It's composed of a mucopeptide frame network. It carries bacterial antigens that are important in their ability to cause the disease. It is stained by Gram-stain and hence classified on two bases, either it's gram positive or gram negative. That will be dealt later on. The lipopolysaccharide on the cell wall of gram negative bacteria account for endotoxic activity. Please keep these things in mind. Now you need to know a difference between the gram positive cell wall and the gram negative cell wall. It very often comes in your theory examination. So please keep this thing in mind. Go through the slide. This chart is also given in your books. You have to remember this whole table as a whole since it comes in your questions. If you prefer to go through the slides, you go through the slides. If you prefer to go through your books, go through your books. But irrespective of what I am telling you today through the slides, you need to go through your books mandatorily. I will be just providing the outline and I will be guiding you how to read this chapter and what points to focus on based on the questions that come in your theory examination. You need to go through the books as a whole. So this is an important slide. This is an important table, mind it. It comes in your question. I am telling it repeatedly. So please get this table by your heart. Now the cytoplasmic membrane. The cytoplasmic membrane is a thin layer lining the inner surface of the cell wall and separating the cell wall from the cytoplasm. It's a semi-permeable membrane. It controls the flow of metabolic compounds to and from the cytoplasm. Chemically, it consists of lipoprotein and small amounts of carbohydrates. About the cytoplasm, bacterial cytoplasm is nothing but a suspension of organic and inorganic solids. It's just like something floating in water. It takes up uniform staining with basic dyes in young culture, but becomes increasingly granular with age. It contains ribosomes, mesosomes, inclusions, and vacuoles. Uh, Definitely, all of you have gone through your biology textbook in class 9, 10, 11, 12. So you know about ribosomes, mesosomes, inclusions, and vacuoles. Well, inclusions you do not know. We will be dealing with these inclusions later on. Definitely, you know about the ribosome. Those are the protein synthetic substances, protein synthesis substances. The mesosomes, these are kind of respiratory organisms, and the vacuoles is just the uh, hollow structures which keep the excretory system, excretory products within them or some other products as per the requirements. So the ribosomes are the centers of protein synthesis. Mesosomes are seen in the vesicular folds within plasma membranes. These are the principal sites of respiratory enzymes in bacteria. It helps in respiration. And the intracytoplasmic inclusions. These are volutin, polysaccharide, lipid, and crystals. Different structures have different functions. Need not know about the details, just keep the things in mind. Which substances are called the intracytoplasmic inclusions? Nucleus. This is the most important part of the bacterial cell. In fact, in all the cell, nucleus is one of the most important part. If there is no nucleus, there is no cell. If there is a nucleus, there is a cell. They may be seen with an electron microscope. They are oval or elongated. Bacterial cell have no nuclear membrane or nucleolus. Actually, within the nucleus, nucleus is a compound structure which composed of two things. One is the nucleolus. This is the most important part of the whole cell in any cell. So it lacks this nucleolus and the nuclear membrane. The bacterial chromosome is a haploid and replicates by means of binary fission. Binary fission means nothing but just a separation of two cells. Slightly telling you a thing in Bengali. আপনারা যারা রুটি করেছেন বা রুটি করা দেখেছেন, জাস্ট রুটি পাকিয়ে রুটি যেরকম করে ছেড়ে, that is a binary fission, nothing else. So they possess either DNA or RNA. It may be either single stranded or double stranded. Depends on the different types of viruses. As we proceed in our topics, you will be getting the examples of viruses which are single stranded DNA, single stranded RNA, double stranded DNA, double stranded RNA, single stranded DNA, etc. etc. In some cases, they possess extra chromosomal genetic materials known as plasmids. Now, these plasmids are the most important parts of a bacterial cell. They has a huge role. Plasmids will be dealt with later on. So, in the classes of microbial pathogenicity, plasmids will be dealt in details. I will be just briefing you about the plasmid. Plasmid is nothing but the nuclear material. In fact, better to say the chromosomal material. Not the total chromosome in a whole, but a segment or a portion of the chromosome. Now, this segment of chromosomes remains outside the nucleus or within the nucleus in a bacterial cell. It's not present in any kind of other cells. It's only present in the bacterial cells. These plasmids contain a specific sequence of genes which are responsible for various important functions of the bacterial cell, which includes antigenicity of the bacterial cells too. Okay. Slime layer and capsule. Many bacteria secrete sticky materials around the cell surface, which is known as the slime layer. Organization of slime layer into definite structure forms a capsule. They may be either polysaccharide or polypeptide in nature. They are demonstrated by negative staining seen as clear halos around the organism against a clear background. So capsules protect the bacteria from lytic enzymes. It contributes to the virulence of the pathogenic bacteria. Now the flagella, another important structure of a bacterial cell. It is responsible for bacterial motility. As we proceed in our topics, you will be getting some bacteria which are motile. You will be getting topics and classes on motility. So motility is an important part of a bacterial cell. With this, the organism moves from one place to another with the help of structures which are known as flagella. Okay, these are Long filamentous structures made up of proteins similar to keratin or myosin that possess antigenic components. I have told you previously that plasmids are which helps in antigenicity of the bacterial cells. Flagella are another structure which helps in antigenicity of the bacterial cells. Antigenicity is nothing but the capability of the bacterial cell to produce a disease. Their arrangements may be either in singly or in tufts, either it may be single or as a cumulative as a whole. It may be located either in a single pole or throughout the body. Depends on the type of the bacteria and their arrangements. Fimbriae are nothing but some shorter versions of flagella which are present all throughout the body. Fimbriae are small filament-like structures present on the body of the bacteria. They are mostly possessed by the gram-negative bacteria arising from the cell membrane. Remember, not from the cell wall. Seen under electron microscopy, they are the organs for attachment. It means that whenever a bacteria is scheduled to show its antigenicity or pathogenicity, the bacteria needs to get attached to that particular organ first. After attaching to the organs, it will exhibit its pathogenicity after getting access into the organs. So the preliminary thing is that the bacteria needs to get attached to a particular cell. This attachment to the particular cell is carried on by the film. So it's nothing but an organ of attachment. Spores. Some bacteria belonging to the genera of Bacillus and Clostridium have the ability to form high resistant resting stages called spores. So some bacteria have some genetic materials within them which they use for their future generations or which helps them in growing themselves. So this is what they do within the spores. They keep these materials within the cell itself in some corners of the cell, which exhibit or which are seen under the microscope as spores. It will be further classified and the arrangement of the spores will be detailedly discussed when we discuss about the topics of bacillus and clostridium. Sporulation helps bacterial survival for long periods under unfavorable conditions. The position of the spores in bacterial cell wall may vary. They might be either oval or spherical. They remain viable for years. They are resistant to drying and relatively resistant to chemicals and heat. Spores germinate only in optimal conditions. Thank you. So this is all about today. Please go through your books definitely. I have selected and focused on those topics which are asked in your theory question papers on a regular basis. So the points I have focused, the topics I have focused, please go through them and keep it in mind and definitely go through your books. If you have any problem, you can definitely ask me. Thank you once again.